Hello everyone, I'm Jason Stockwell from the University of Vermont. I'll be presenting the efforts of our working group called GEISHA, which is examining the impacts of storms on lake phytoplankton communities. This wind event on Lake Champlain and its impact on water column thermal structure is a great template for thinking about how storms may impact phytoplankton. Species adapted to a stable water column and its associated conditions may do well before the event, but after the event, opportunities may exist for colonizing species to gain a foothold. Our goal is to assess when and under what conditions storms may result in phytoplankton community shifts and how such shifts are related to changes in the lake physical state. Our goal is to provide a new framework to explore theoretical questions on species diversity and succession in aquatic ecosystems. GEISHA is actually a sub-project within the GLEON Project Storm-Blitz, which started in 2013. GLEON, or the Global Lake Ecological Observation Network, is a grassroots organization of many people from many different disciplines interested in conducting research on the role and response of lakes to climate change. Much of the focus has been on using high-frequency data from sensors deployed in lakes around the world, although GLEON efforts also rely on traditional long-term data sets. A key feature of GLEON is sharing of ideas, data, and research efforts. Reynolds showed that in the absence of external disturbances, phytoplankton communities would go through true succession with seasonal changes in their physical environment. We are interested in how storms may alter the physical states of lakes, how this might result in community change, and if there is a phenology to impacts depending on the timing of storms. Following the GLEON approach, we look to existing data sets to address our questions. Several of us spent time on Storm-Blitz over the next year, presented posters at GLEON 16, but realized, as usual, funding was needed to move Storm-Blitz forward in a meaningful way. Eventually, we were co-funded by the Center for Synthesis and Analysis of Biodiversity and the USGS John Wesley Powell Center to support the efforts of a smaller, focused group. We were very fortunate that the two directors had similar interests and were willing to engage in and explore options for co-funding. CESAB typically funds 6 workshops plus a postdoc, and the John Wesley Powell Center typically funds 2 to 3 workshops plus a fellow. Each agency saved money and made their dollars stretch. Additionally, this was the first time each had ventured into something like this together, and thus was beneficial in other ways than just purely financial. We have also been fortunate to secure supplemental funding from the Vermont Water Resources and Lake Study Center to partially support a string of postdocs over the last few years. We have common tasks similar to any working group, including gathering the data, developing our methods, running the analyses, and synthesizing the results to push the science forward. We'll dive a bit deeper into this in a little bit. The core group is highly international, bringing people together from many different disciplines, years of experience, and backgrounds. Additionally, we have many involved beyond the core group, including those who are generously contributing data and those who are interested but did not participate in the workshops. How do you work together effectively with so many people involved from all over the place with different levels of involvement? Some of the challenges are technical in nature, others are non-technical. For example, we have data sharing agreements, data templates, data management plans, authorship policies, and so on, and email and Zoom to help with the nuts and bolts of the project. These are the technical solutions. These approaches go a long way, particularly when working outside of the core group with, say, the data providers. Equally important are the non-technical ways in which we work together. What are the experiences and expectations we bring to the table? How do you build trust among a group of people entering a series of workshops who may not all know each other? Here I'll focus on our core group, which met for the first time in December 2016. Going into the first workshop, we took the perspective that GEISHA was a marathon, not a sprint, and that spending significant time on team science concepts up front would pay big dividends in the long run. Much of the first 2 days was spent getting to know each other, having discussions and presentations on many aspects of team science, including past experiences, expectations, working with others, communication, and so on. We also took time to discuss conflict, that we could expect to have disagreements, and that this is a normal process when people collaborate. There is this groan zone that occurs between idea generation and decision making. Many of the participants commented that this was their first time in a working group where deliberate time and effort was spent to help everyone communicate better, and were very appreciative of the approach we took. In terms of actual science, we held 6 workshops with the core group from 2016 to 2019. To date, we have compiled data from 40 lakes, generated an R package, and produced several manuscripts at various stages of production, which I will highlight in a little bit. An important point, and one that the director of CESAB pointed out during our last workshop, was that it takes time to get all our ducks in a row, and that the point where you are ready to dive into analyses and write manuscripts is usually the point where the funding for your workshop ends. As you'll see by the end of this talk, we are really in the middle of our 3rd and 4th tasks. As I just mentioned, we have data from 40 lakes and reservoirs. Traditional longer-term surveys make up the bulk of our data. Our criteria include a sampling frequency of at least every 2 weeks, phytoplankton taxonomy down to genus level, vertical profiles of at least temperature and DO, and weather data. We have a minimum time period of 5 years, although this is flexible. We are also very interested in concurrent high-frequency buoy data when available. As I'll show in the next slide, combining the data from the 2 different sampling frequencies will likely prove very important. Our primary data challenge revolves around taxonomy and timescales. First, phytoplankton taxonomy is just inherently difficult. We need to account for different and changing skill levels among data sources, methods, and so on. I'll speak more on this in the following slides. Second, depending on time scale of storm impacts, our biweekly sampling frequency may miss any shorter-term effects. For example, if we have some baseline condition that is disrupted by a storm, a quick but short response may go undetected by our 2-week period. However, if the impact is longer in duration, then our 2-week sampling frequency may be sufficient. For this reason, we expect those lakes where we can couple both low and high frequency datasets to provide valuable insights. With data from so many lakes and across so much time, some data going back to the 1970s, we had to first standardize across the more than 3,000 unique taxa names for different spellings. Additionally, phytoplankton taxonomy is continually changing, so it was necessary to standardize across lakes in time if we wanted to have a common taxonomy. We checked all names against algaebase.org, and if we found an updated name, as in this case, we used that taxonomically accepted name. In this way, we ensured our taxonomy was constant across all our data sets. We also classified our species into functional groups, where species share certain physiological, structural, and ecological traits related to their function in the environment. Many types of classification systems are available and range in their criteria for the functional classifications. Perhaps one of the most widely used functional groups system in ecology is R and K selected strategies based on growth. The morphologically based functional group shown here is based on size, shape, and structural characters. Application of functional groups to species data overcomes some practical limitations, including variability among labs in identifying samples down to species and reducing the number of zero observations to remove constraints on types of statistics and modeling one can use. Finally, we can ask questions through lenses. Do we see different patterns when we examine data at different levels of functional taxonomic organization? We developed an R package that can take species lists of phytoplankton and convert them to functional groups. In particular, species are assigned to their genus, to their morphofunctional group, and to their CSR group, so we can easily apply analyses to different organizational levels. We just published an invited review paper in Global Change Biology, which provides a conceptual framework for how storms may influence phytoplankton dynamics. At the heart of the framework is how functional groups respond to their environmental conditions. Groups with certain traits do better in certain conditions. For example, we can expect flagellated taxa that are mobile are expected to do well in stable stratified environments, but not so well in turbulent environments, whereas the opposite is expected for taxa with small sizes and rapid growth. If we combine these functional traits with how weather and lake watershed attributes interact to influence lake conditions, we then may be able to predict how phytoplankton communities may respond to storm events as mediated by lake and watershed attributes. For example, we might expect heavy rain to reduce light availability in a lake that has high watershed to lake area ratios, small lake volume, and developed land, whereas light availability may not be affected by the same rainstorm in an undeveloped lake with a low watershed to lake ratio. One of the main points of our paper is that we can't expect all storms to have the same impact on lake systems, and thus on phytoplankton. Storms are mediated by the watershed, the lake, and the pre-existing conditions in both. Storms directly influence lakes by imparting their energy to generate, for example, mixing, but the degree of mixing is related to the fetch of the lake depth, and how strongly stratified the lake is at the time of the storm. Storms indirectly influence lakes as watershed characteristics such as slope, composition, and soil moisture content will mediate the delivery of runoff to the lake as well as the content of that runoff. Both of these filters will influence how lake conditions change as a result of a storm, and thus influence how phytoplankton communities are able to respond to any changes. We finished the review paper with 6 areas we believe are leading research directions. I won't cover them here, but perhaps the most interesting one to think about is our perception of what storm is versus what a storm is to a phytoplankton. What we see and experience above the surface may not be the same level of disturbance below the surface. We introduced the concept of a limnological storm versus a meteorological storm to start framing how we can view storm impacts through different lenses. In other work, we have a manuscript in revision that examines how storms may alter lake surface temperatures. We know that temperature is an important variable for phytoplankton growth, so we used our data sets to ask to what degree do storm events alter epilimnetic water temperatures and thus could shape phytoplankton dynamics. We expected that storms would be characterized by large increases in wind speed and large drops in epilimnetic temperature. So in plots of change in wind speed versus change in water temperature, we expected to see a cluster of points in the lower right-hand quadrant. When we use daily data from 17 lakes, we see that the largest increases in wind speed do not produce the largest decreases in water temperature in most cases. The axes are the same as in our conceptual figure, and the colors represent surface water temperatures to tease apart seasons. We found that the top 5% increases in wind speeds had an average decrease of only -2 degrees Celsius, and the maximum decreases for such wind events were typically less than 2 degrees Celsius. Results were similar for the same analyses looking at rain events. What we conclude is that the storm impacts on phytoplankton communities are likely not going to be manifested by temperature changes, and perhaps changes in nutrients and light availability may be more important. Another interesting question is, how do we know if a phytoplankton community changes after a storm, whether it is a result of the storm or if the change would have happened anyway? We expect storms to revert communities back to earlier successional stages, but this may be modified by lake type, for example, shallow versus deep lakes, or by season. One could actually expect succession to be sped up in the fall if a storm breaks down a stratified system, for example. We are using the data set to estimate what an average seasonal trajectory looks like in a multivariate community space, and then identify those cases where the community trajectory went outside of the normal trajectory. We have 8 data sets from large lakes, and the highlighted areas show the maturation process of each community from spring through summer. You'll see Topo on the left looks a bit different because it is in the southern hemisphere. The shortest maturation seasons are in the highest latitude lakes such as Vuosaari in Finland, and the longest maturation seasons are in the lower latitudes such as Kinneret in Israel. Preliminary results suggest storm-induced reversions are highly variable across lakes, and that reversions occur in the absence of storms. When the changes in physical conditions of lakes during reversions are compared with and without storms, the intensity of those changes are no different, suggesting other mechanisms. Perhaps an even more interesting result is that detection of storm-induced reversions appeared related to the length of the time series and the number of storms. These methodological issues are something we are working on. At a different scale, we are asking whether phytoplankton biodiversity is related to the level of disturbance lakes experience from weather. In this conceptual figure, we are going to plot 2 measures of wind. One is how often it is windy on the x-axis, and the other is the strength of the wind on the y-axis. In the lower left, we have low wind conditions. It is not windy often, and when it is windy, the wind is not that strong. Here we expect low phytoplankton diversity dominated by stress-tolerant or competitive groups under the CSR functional grouping. In the upper right, we have lots of wind and lots of strong wind. In this case, we might expect higher diversity, but if the disturbance is nonstop, we might expect that to decrease. Here, the ruderal, or disturbance-tolerant group would dominate. In the upper left, we have relatively little wind, but when the wind blows, it is fierce. This quadrant would be low diversity with the occasional increase as a result of wind disturbance. And in the lower right, we have constant wind, but at a lower magnitude. Here we might expect higher diversity with competitive groups dominant. The intermediate disturbance hypothesis suggests that moving through these various wind quadrants may maximize biodiversity. Conditions are not so stable that one dominant species or group emerges through competitive exclusion, and conditions are not so disturbed that the environment is hostile to most. Here we plot the data for a number of lakes for just 1 year of data, and a threshold of 2 meters per second as our threshold for counting a day as windy. Here we can see 3 lakes where the wind is relatively constant and strong in the upper right-hand corner, whereas the remaining lakes are relatively less windy and the magnitude of the wind is middling to low. The color of the circle and lake names and the size of the circle represent species richness over the year. And according to the intermediate disturbance hypothesis, the largest and most green circles should be in the middle area. This is an idea we just developed, and it still needs a lot more work, and fortunately we have a lot more data. In this last stretch of the talk, I'm going to transition to our data and its availability. We are in the process of uploading the data to UVM's Forest Ecosystem Monitoring Cooperative website, where you can find the project under the search GEISHA. We have time series of data from 40 lakes, and each lake will have all or a subset of the following data: high frequency, weather, and lake data. We typically provide daily averages for when data are collected at high resolutions or higher frequencies, and sometimes we will also include the raw data. Long-term data for lake physical and chemical conditions and for phytoplankton. These data are typically every 2 weeks, but in some cases they are weekly, and in one case the data are monthly. We also provide derived data on lake physics. These data are a result of combining weather data with the lake thermal profiles to estimate stability indices, thermocline and mixing depths, and so on. For each lake, we include metadata in a QA/QC notes document that identifies any issues with the data set, how we remedied those issues, and every update to the data sets. In some cases, data providers have given us permission to post all of their data, and in other cases, we can only post a subset of the data sets. In all cases, we provide the data provider's contacts such that anyone can request any data that are not made public. Here is an example of what you will see when you click on datasets for GEISHA. If you click on the Acton Daily High Frequency Lake data, you will see the data structure and you will be able to download different parts of the datasets or all of the data. At this time, we have all the data for Acton Lake in Ohio uploaded and available, and we will be adding new datasets as fast as we can over the next 6 months. To conclude, I hope I've given you an overview of this fairly unique dataset that our group has been working on over the last several years. We think it will contribute to better understanding of the impact of storms on phytoplankton as mediated by watershed and lake attributes. The large number of lakes in the dataset will allow us to examine such questions across lake types and environments. We also hope that the background information about our process for working both closely and loosely with a large network of collaborators has provided insights on team science in practice.